అక్కడ నెట్వర్క్ చెక్ చేసుకొని వస్తున్నారు. 11:05 వరకు స్టార్ట్ చేస్తున్నారు. 1:05 Good morning to all. Good morning, students. YouTube live and Facebook live is also coming. Please check it and join there also. Students, please inform to your friends who has not joined. Let them join in YouTube and Facebook also. Once I will call to Sir and let him also join as early as possible. Sorry for the delay today. Can you hear me? Please respond. My voice is audible. Please respond in the chat box. My voice is audible or not? Please respond, students. Yeah, students, I had called to Sir. Within 1 minute, Sir will join. Please inform to your friends also who has not joined. Let them join as early as possible. The section will be start at 11:05 sharp. Okay.
Good morning, Maheshwari. Can you hear me? Maheshwari, can you hear me?
Good morning, sir.
Very good morning. Let me just share the screen.
Yeah, please on your video also.
Yes,
Your voice is good today, and if you want PPT and camera means I will check and I will inform you.
please. Yeah. Hope
Yeah,
my
okay.
video. Yeah,
Yeah,
video is
your
audible.
video also good, and PPT.
Yeah, I'm just sharing the screen. Is my
Yeah,
screen visible?
yeah, it is visible, sir. You can continue.
All right, fantastic. So today we will have a quick class on a further topic on drug discovery development. So first, let us review and revise what we have done yesterday. So very good morning to all of you, and thanks for joining this class. Today I'm in much warm clothes because it's real chilling cold out here with 3 degrees temperature. So I'm sure all of you must be also enjoying the cold climate this year. So all right, let's get started. Maheshwari, just confirm me once my slides are visible, my audio is okay, so that I can get started.
Yeah, it is good, sir. You can continue.
Thank you. So yesterday we had talked about pharmacokinetic studies. So I don't want to get into the entire revision details, but still, I just thought we'll cover a few highlights of that, and then a few more points we can discuss today. But the idea here is to just revise ADME. In fact, I was just thinking that we can revise. After this is over, we will quickly, in a snapshot view, we will revise the things which we have done, even in the prior classes also today. Okay, so we yesterday talked about the principles of ADME: absorption, distribution, metabolism, and excretion. We talked about pharmacokinetic parameters. We discussed how the parameters are calculated. We talked about how do we really interpret preclinical PK parameters, what is the importance of each of those parameters. Yeah. So as we started yesterday, that there was a time almost a decade ago when used to fail because of poor pharmacokinetic properties, and pharmacokinetic properties absorption, distribution, metabolism, or excretion, any properties used to be one of the major reasons for failure of drugs in early drug discovery. So attrition of drug used to happen because of that. But that is almost a decade ago. Now our understanding on PK principles and the importance of PK is improved. I mean, has improved overall, and also because of advancement in technology. Now, what is happening is this attrition, because of poor PK properties, has come down to almost have come down to less than 10%. So that's the difference between 2000 and 2020. Okay, so DMPK plays a major role, and DMPK scientists play a major role whenever PK comes into the play, because whatever data we generate goes back to the chemist, and they can really modify their structures in order to optimize the PK properties of a drug. Same way, sometimes like solubility and permeability, even the formulation scientist, they can also benefit from this data, and they can really modify the formulation in order to improve the solubility or improve the permeability of a compound. So that's the role of pharmacokinetics in drug discovery and development. This is just an example. We have discussed this thing in detail. Pharmacokinetics. Now, I'm sure all of you know what body does to the drug. That is, body is absorbing the drug. distributes the drug, it metabolizes the drug, and finally it excretes the drug outside the body. That is called PK. So what body does to the drug is pharmacokinetics, and what drug does to the body, the pharmacological effect which is elicited by the drug on our body, is called pharmacodynamics. So what the drug does to the body is pharmacodynamics. Then we talked about the drug. The moment it reaches the systemic circulation, it can either reach liver, can get metabolized and then get eliminated from the body through kidneys, or it can directly go to the kidney and get excreted. From systemic circulation, it can reach to different tissues where it gets distributed. So the parameter called VD, volume of distribution, is basically derived deciding the distribution of the drug in the body in different organs and tissues. Whereas excretion and metabolism, they are responsible for clearance of the drug. So that's why clearance and volume of distribution are defined by the distribution of drug to the tissues, and excretion and metabolism from the body for the clearance. So what are the primary PK parameters? It's clearance and its volume of distribution. What are the secondary parameters? Half-life and bioavailability. Then we got into details of each of the parameters yesterday, starting from clearance. As we discussed, clearance is nothing but how efficiently or rapidly a drug is eliminated from the body. And when you say elimination, it comprises of metabolism, which can happen in liver, which can happen in intestine, lung, or kidneys, and excretion, that is through urine, which is a major source, then via bile, saliva, milk, and other things. So basically metabolism and excretion together defines the elimination. So clearance, how it is defined, it is the rate of elimination by blood or plasma concentration. So how do you actually practically calculate clearance? That is, suppose IV clearance, intravenous dose. So dose, when you divide that by area under the curve, now all of you know what is area under the curve. So dose by AUC, that gives you clearance. If it is oral route, you do dose by AUC oral. But as we have discussed, please remember it's apparent clearance, because in IV it is 100% delivery of the drug, whereas in oral it is defined by absorption. So you have to divide that by F, which is bioavailability. Only then you get accurate clearance after oral administration. Just by pictorial diagram, if you can see the green one, which is a low clearing compound, so lower the clearance, longer is the half-life. The red one, it is high clearance, so it is a steep getting cleared within 60 hours. So shorter, so higher the clearance, shorter is the half-life. Lower the clearance, longer is the half-life. So it says 10 ml versus 2 ml clearance per minute per kg. So green one was 2 ml low clearing compound. This was high clearing compound. So the relationship with the half-life for clearance is inverse proportional. So clearance is inversely proportional to half-life. Okay. Then we also discussed about importance of blood flow. So like each organ have different blood flow, while species comparison, when we are trying to do, say for liver, in different animals, the blood flow is different, right? So for mouse, it is 90, for rat, it is 65, for monkey, it is 44. So then we talked of something called as extraction ratio, ER calculation. So for any new compound, it can be done as long as clearance is known to you. You can find out the extraction ratio. How do you do it? You do clearance, what is given for the NCE, which you calculate. You divide it by the organ blood flow in liver, especially in that species. In this example, it was monkey, so it was 44. So 25 divided by 44 is 0.56. So practically always remember, it says that 0.3 and 0.7. So whenever you find extraction ratio less than 0.3, means it is low clearing compound. The extraction ratio of between 0.3 and 0.7 is intermediate clearance, and any extraction ratio value which you get above 0.7 means the compound is high clearing compound. So this information will really tell you to interpret the data. Suppose 5 different compounds you've done a PK study, and based on the clearance you want to rank order them. You can actually do that using this extraction ratio formula. Then the relationship of the total clearance with hepatic and renal. So you can find renal clearance, that is amount of drug in urine multiplied by the clearance value which you got becomes renal clearance. When you subtract this from the total clearance, you get hepatic clearance. So basically hepatic clearance from where do we get? When we do in vitro assays, that is using microsomes, using hepatocytes in detail we have talked about, you can always find hepatic clearance. Now, with our improved discussion and understanding on enterocytes, in this we can also add up the clearance from intestine as well, because that fraction is also now contributing. So that will become the total clearance. Then we talked about volume of distribution. So volume of distribution on basically it is like an apparent volume of distribution. We call this as this is never considered as an accurate volume of distribution. It is a mathematical assumption basically. So that's why we call it as apparent volume of distribution. So for in human, say for example, the total body water for a healthy human of 70 kg is around 42 liters. Then we really find this apparent volume because why do we call it apparent volume? Basically, it is defined by amount of drug in the body divided by blood or plasma concentrations. So as practically it is just next to impossible to find accurate exact amount of drug in the entire body, so we call it as an apparent volume, right? So because it relates to the blood or plasma concentration of a drug. Okay, so from an example, very simple example of a container where 10 ml was taken and we added 5 mg of the drug, so you can find the measured concentration is 0.5 mg per liter. If I had to back calculate the volume, then it comes 10 liter because it is 0.5 mg per liter. Same thing if I add glass beads into the same container, the drug will get adhered to it. Now if I am trying to find the concentration from a sample, it will be diluted. Say it comes 0.05 mg per liter on analyzing the concentration. If I back calculate how much liters of water in total, so it comes to now 100 liters. So that's why we call this as apparent volume of distribution. Just an mathematical model to understand that. But let us understand the role of actually volume of distribution from a physiological perspective. Please remember, whenever you have T half change is directly proportional to volume of distribution. So what does it mean is, like you have high volume of distribution here for the red graph, which is 5 liter per kg. So as you can see, the half life is also going to be longer because the compound is lingering around in the body all the way till 24 hours. So naturally half life is going to be longer for this. But whereas for the green one, where the volume of distribution is 1 liter, you can see half-life is also going to be shorter. How do you get half-life? Suppose the total dose was 100 mg. By the time you get half the dose remaining in your body, that is the half-life. So it is directly proportional to half-life. So for the like we have seen the ratio extraction ratio for clearance, for volume of distribution, we say that if you have volume of distribution at steady state less than 0.3 liters per kg, it is considered to be small volume of distribution. What does it indicate or imply? That there could be high protein binding in plasma and it does not distribute to the tissues. So why I mean, where volume of distribution, low volume of distribution is applicable, that is for vascular or extracellular targets. For intracellular targets, the application is higher volume of distribution. So when you get anything around above 0.7 liters per kg, means it is having relatively large volume of distribution. What does it mean? It indicates that drug is distributed outside the vascular space. Okay, so that's the meaning of the volume of distribution and its role. Then we moved on to half-life. So half-life, as we have seen, is defined as time taken for the concentration of the drug in blood or plasma to decline to half of its original value. So as I gave that example, you're taken 100 mg dose, when in your body there's 50 mg remaining, that time the time it takes to reach to half the concentration of the original one, that time period is called half-life. So T half is a hybrid pharmacokinetic parameter. It is determined by two parameters, which we just now talked about: clearance and volume of distribution. And in the formula also, it is pretty straightforward. It is directly proportional to volume of distribution. So T half is volume of distribution into elimination rate constant, which is 0.693, divided by clearance, because as we already discussed, clearance is inversely proportional to T half. So T half is 0.693 into volume of distribution divided by clearance. Higher the volume of distribution, higher is the half-life. Lower is the clearance, higher is the T half. Higher is the clearance, lower is the T half. That is their relationship. So why useful? In fact, yesterday also a couple of questions were there, that why clearance is useful, why half-life is important. So this is the answer, that basically if you have a longer half-life, the dosage regimen can be decided. Suppose 10 to 20 hours half-life is there, means once a day dose is possible. Many a times for long chronic disease, you take medicine once a day, because the half-life is longer, so you can afford to take it once a day. For antibiotics, generally the half-life is shorter. So what do? We take 2 to 3 doses a day. So that's the reason why half-life is very important. Based on that, when you want to really do multiple regimen, multiple dose has to be given. You can decide the dosage interval, when you want to repeat that. As we have taken the example yesterday of half-life for antibiotics, you would always want that your concentration should be above MIC, which is minimum inhibitory concentration for a specific microbe or a bacteria you are trying to capture or trying to elicit effect against. So for an antibiotic, we always want that our blood concentration should be above the MIC. So this half-life will always give you an advantage or an implication when exactly you should repeat the dose in order to maintain consistently blood concentrations above MIC, so that antibiotic can be highly effective. So that is the role and importance of half-life. There are some disease indications like neurological disorders where half-life of the compounds are even 100 hours. So once in 2 days, once in 3 days dosing is also possible. So that is the importance and role of half-life. Again, it is an indication of decline in plasma concentration over time. Okay, so this is just an example we have seen. So based on this, you can actually decide the half-life, and then you can choose which drug you want to take. In this particular example, the one with a blue curve is more preferable, although half-life is same. Why? Because with this half-life also, if you go by the concentration, this particular black dotted line is the efficacious concentration. So this A drug is more preferred. Why? Because although half-life is same, the concentration over a period of time is well above the MIC for, or well above the efficacious concentration for drug A. That's why A is more preferred over B. So you get the point. To decide which particular drug also to use, or when you have a choice of drugs to be used, you can also decide this way. In this case, half-life is same, but we have gone by the effective concentration in the blood. Based on that, we have decided which is the better drug. Then we moved on to one of the very important factor, parameter, PK parameter, that is bioavailability. So bioavailability, again, the drug when you take it is absorbed. After absorption through intestinal barrier, it crosses that through portal vein, it reaches to systemic circulation after liver. So from systemic circulation, it gets distributed into the tissues, and from liver, it gets metabolized and excreted through urine, right? So for all this absorption kinetics, one of the important parameter is bioavailability. So bioavailability takes into account area under the curve. Okay, so area under the curve and absorption rate constant, and other parameters related to this are Cmax and Tmax. So these four parameters also we have understood yesterday and discussed. Let's see what it is. So as I said, in order to find bioavailability, which is nothing but amount of drug available at the site of action is called bioavailability. So when you want to find a bioavailability after oral dose, okay, you should have one study done using intravenous route. Why? Because as we have discussed earlier, in intravenous, entire drug amount we are delivering directly into the circulation. So assumption is it is 100% delivered into the systemic circulation. So bioavailability is 100% following intravenous route. So same thing, I have to find the bioavailability for oral route. How do I do it? So you practically have to compare the area under the curve after oral administration with area under the curve after intravenous route. So this basically tells you how exactly you can do the bioavailability calculation for oral route. Correct. So as you can see, just one second. I think I'm here. I have to open it up here. Yes. So basically, just it gives me an idea how exactly you can calculate the bioavailability for an oral route. So the formula for that is PO is for AUC oral route divided by dose of oral route. When you divide this by AUC IV by dose IV, so this will actually give you the bioavailability after oral administration, right? We can also use bioavailability calculation when you want to do what I call comparison of different dosage, like different relative bioavailability, say between tablet and a suspension. So even that, it can even in that case, it can help you identify which is better in terms of bioavailability. And how do you do it? In solution, we consider it as 100%. So when you compare a tablet versus solution, you will come to know what is the relative bioavailability between these two dosage forms. That is tablet versus solution. Okay. So the major factor which plays a role in calculation of bioavailability is area under the curve. We will understand in subsequent. I mean, of course, we have discussed yesterday that how exactly AUC is calculated. Okay.
Excuse me, sir. Can
Yes.
you put full screen?
Okay. One second.
Not cam. PPT full screen is not coming. Yeah.
Is it there?
Okay, sir. Yeah, okay.
Right. Yeah. So as you can see, we have just taken an example of bioavailability of a solution versus suspension. So in this case, it's clearly visible the one with the lowest area under the curve, because this is the curve. It is basically it's an IV. Okay, when you do it with a solution, you're getting a higher area under the curve, and same thing when you're doing using a suspension, which is again less compared to solution. So this the if I connect each of these dots to the x-axis, it will form a trapezoid. So under one curve, under different time points, whatever trapezoid number of trapezoid it is forming, like this, you have to total the area of each of that trapezoids, then you arrive at total area under the curve. There are two ways they are reported. One is AUC 0 to infinity, which is an extrapolated value, and another is AUC 0 to say time point. In this case, say suppose it is 24 hours. So area under the curve 0 to 24 hours is nothing but whatever trapezoid it is forming till 24 hours of time point. Summation of areas of all those trapezoid will give you AUC 0 to 24 hours, and from that another extrapolated value we can get is AUC 0 to infinity. Okay, so then very quickly we talked about protein binding. So protein binding, as I said, that drugs which are, I mean, the free drug is responsible to elicit a pharmacological action, and free drug is the only drug which will really reach the site of action basically, and bound drug is there in plasma, because in plasma there are proteins where a drug can be in a bound form. Like in tissues, there is a non-specific binding to the proteins. In plasma, there are proteins like albumin, which are those proteins. So alpha-1 acid glycoprotein, lipoproteins, and albumins. To this, it gets bound. So always remember, it is a free drug which we look for pharmacological action. Multiple dose regimen, we already have talked that for therapies like chronic dosing are required on a fixed or a fixed time interval to maintain a consistent drug exposure. So you may have to do multiple dose regimen also. Preclinically, animal efficacy is often demonstrated through repeat dosing. So, of course, in subsequent lectures, we are going to talk in detail about the efficacy models, where we will discuss in detail about how exactly common efficacy models are utilized. Say for diabetes, we have a model called streptozotocin induced diabetes, alloxin induced diabetes. So for all these models, we have to do repeated dosing, right? Because we want a certain concentration to be maintained in our body. So we will understand that as per the model wise when we study about efficacy models. Like same way we do toxicity studies. So even for toxicity studies, in animal toxicity studies, we have to do repeated dosing. So that really defines the multiple dose regimen in animal toxicity studies. Okay, so as it is said, before conducting repeat dose studies, safety assessment after multiple dosing in preclinical animal species is required. So we will also be talking in detail about toxicity studies, where we'll talk about single dose studies. We'll talk about repeated dose toxicity studies like 28-day toxicity studies, where daily doses are given up to 28 days. Then 90-day toxicity study, where up to 90 days you have to dose the animals at different dose group, and then we sacrifice and try to look for the toxicity if at all it is observed in any of the organs. And then there are long-term 6 months and 1 year carcinogenicity studies, where repeated dosing has to be done. So that is the application of multiple dose regimen. Why do we do this? Is because our target disease, say for example, is diabetes. So target species is human. So as you can imagine, for diabetes type 2 or type 1, these are long-term treatments, 6 months, 1 year, or years together. So when you want to use drug for such a long period, you would like to know if they are safe, right? So in order to establish the safety of your drug, we may have to do repeated dose toxicity study. We may not do for years together, but at least for a long-term safety studies we have to do. So that's where 28-day, 90-day, 6 months toxicity studies are there as per the guidelines. So once you generate that data, then we can extrapolate that safety into human as well. Okay, and then key parameters in multiple dose regimens. What are the key parameters? So I was always talking about volume of distribution at steady state. So what is steady state? So this is basically after certain multiple dosage, we attain a stage where there will be consistent concentration maintained over a period of time. So that when you reach that stage, that is called steady state concentration. Okay, so it depends on dosing rate and the clearance of the compound. So this is defined basically by these two PK parameters. So this steady state is nothing but it is a time to reach the plateau. So based on the half-life, you will decide the dosage frequency, and over a period of time, it will achieve it will attain a certain plateau concentrations, that is called steady state. Okay. So, in nutshell, what we have discussed and understood about ADMA parameters is clearance and volume of distribution are primary PK parameters, which are very important to optimize the molecules early in drug discovery. And half-life and oral bioavailability are equally important, because bioavailability will define the exposure of the drug, and half-life will really help you to decide the dosage regimen, the frequency of dosing also. These are secondary parameters but equally important as clearance and volume of distribution. And bioavailability, we take into account one parameter, which is area under the curve. Okay, so area under the curve can be compared against a oral, against a IV, and you can calculate bioavailability. As we have discussed, that when you do the studies in vivo early in discovery, if at all you want to make some changes in your structure to in order to optimize this parameter, it is doable. So you can go back to the chemist, you can share with him that it is showing high clearance, maybe because of high metabolism, because as we have discussed, clearance is defined by excretion and metabolism. So if you go back to the chemist, to a chemist, and say that your structure you have to modify in such a way that efficacy is not changed, but the stability is added to my structure, in the sense it is getting easily metabolized, so that it is getting easily cleared from the body. If somehow you can make it more stable so that it takes longer time to come out of the body, so clearance would be then slow and my half life will also be accordingly longer. So that can be done using structural modification in consultation with a chemist. So that's why preclinical studies in discovery, it really helps to optimize pharmacokinetic profile of a drug candidate. At the same time, when a chemist says that no further structural modifications are possible without altering the potency of the molecule or the safety of the molecule, you can always work closely with a formulation analytical scientist and somehow make your formulation in such a way where you can improve the solubility or permeability and other parameters which you can play around with in order to optimize your molecule. So that is the importance of pharmacokinetic studies in drug discovery. Okay, so this is just an example pictorial presentation. Like you are giving, it is reaching the peak, coming down. Again, you are dosing them. So keep on dosing. You see, at some point in time, a plateau has reached. So now, no further increment in the concentration. It has reached the plateau. So this is what we call as steady state. Got it? Then there is something called as dose strategy. So drugs which are having high volume of distribution, they are actually given by this strategy, because high volume of distribution means more drug is getting distributed to the tissues. So less of the drug is available for action in the circulation. So this can be given in this fashion. So first you can give say a large dose, which generally we call it as loading dose. Okay, why do we give that? Is at that first dose itself you try and attain the steady state quickly. Okay, and then later on, to maintain the plasma concentration, you may give maintenance dose. So that is the concept, that first give a loading dose, a large dose, in order to achieve the steady state faster. If I go back here, so the idea here is give more dose so that drug can reach in steady state. Once it has reached here, there's no point in giving more dose. Just give them maintenance dose, which is definitely less than the loading dose, just to maintain the drug concentration in this range of steady state. So that is the strategy being taken. How do you calculate that? As I was telling you, volume of distribution plays a role in this. So loading dose, how do you calculate is volume of distribution into target plasma concentration. So this is how loading dose is calculated. So when students are doing their dissertation work and research, or whenever they do any PK study, you can always, after doing that, you can actually you can do that at any dose what you have decided, a PK study. But when you want to recommend a dose, an efficacious dose for any species, you can always recommend, calculate the loading dose and the maintenance dose, and you can recommend in your thesis, because this is how loading dose and maintenance dose are calculated, and this is the this information you can definitely include in your dissertation or thesis work. Okay, so maintenance dose, see, when you do a dosing, you want exposure, correct? And maintenance dose is defined by clearance. So based on the clearance, you will define how much dose needs to be given in order to maintain in a steady state. So that's why maintenance dose, to calculate that, you have to multiply it by clearance. So here, loading dose is volume of distribution into target plasma concentration, whereas maintenance is clearance into target plasma concentration. Okay, So there is something called as therapeutic drug monitoring. Okay, so it's a process by which dose of the drug is adjusted according to its plasma concentration. Okay, which the drugs which are having wide variation in the PK. Okay, there is a lot of variability in inter-individual versus intra, like for different, you're taken six individuals, and within the individual data there's a lot of variability. Then you actually do the therapeutic drug monitoring. Okay, so how why it is done for those whose effect cannot be easily measured. Say for example, an anti-hypertensive drug, it can be easily measured by monitoring the BP. Okay, so TDM is not used. So any drug where you can measure the effect easily, TDM may not be required. Where it is not, you have to do therapeutic drug monitoring. So there is a concept called fixed dose ratio combination. So why do we do this? The advantages, the convenience, and better patients compliance. Like, so there are certain symbiotic synergistic drug combinations which are being used, like sulfomethazole and trimethoprim, levodopa plus carbidopa or benzazide. So combination, these are all oral contraceptive combinations are also available. So sometimes you have to use fixed dose, but in a combination way. Sometimes therapeutic effect of two components may also add up, like amlodipine and atenolol. Like there is a over-the-counter drug available, say telmisartan. So telmisartan and amlopidine, these are amloperidone. These are both used together to elicit extra effect, right? So it is Telma AM, the drug which is available in the market. Say for diabetes, many people take the safest drug is metformin, but sometimes when you feel that the glucose is not coming under control, we use it in a combination, which is metformin with glimepiride. So now there are different combinations. You can use 1 mg glimepiride and 500 mg of metformin. You can use 1000 mg and 2 mg of glimepiride. So like that synergistic effect, because both are anti-diabetic drug, both bring down the glucose concentration. Okay, sometimes you have to use this combination because side effect of one component needs to be counteracted with the other one, right? A thiazide and a potassium sparing diuretics. So this is a combination where side effect needs to be nullified. At the same time, there are diseases where single drug may not be useful. Like for TB, there is a huge issue of resistance. So when you look for the tuberculosis treatment regimen, it is always a combination of 5 to 6 drugs, right? So fixed dose ratio, combination preparation. This is the concept. Sometimes for synergistic, better pharmacological action. Sometimes to counteract
Sir,
the effect.
excuse me,
Yes, yes, ma'am.
sir. Sir, from your backside, some lady's voice is disturbing.
Is it?
Some ladies are talking, I think, at your background.
Uh, I think it is two. It is after the two houses there down the line, but I don't know. One second, let me. Can I just take 1 minute to sort it out?
Yeah, okay, sir. ఇప్పుడు ఇచ్చారా ఎప్పుడు ఇచ్చారు అదే ఎప్పుడు ఇచ్చారు కప్పు మరి ఇప్పుడు ఇచ్చినట్టు చెప్తున్నావే ఆ, అదిగో టెన్షన్లు ఎక్కువైపోయినాయి మేడం బరువు బాధ్యతలు ఎక్కువైపోయినాయి వెంకటేష్ గారికి Okay, madam. రాలేదు అభిలాష్ రానా? Sorry, students. Sorry for the inconvenience. Sir facing some issue. I have informed to sir. Within 2 minutes, the section will be.
Manishwari, can you hear me? Hello?
Yes, sir. I can hear you.
Okay, I'll just share my slide.
Yeah. Okay.
All right. Can you see my slides now?
No, sir.
Can you see now?
No, sir.
It is showing.
It is black screen.
Uh,
Can you once again stop the share?
yes, I will do it. I will just do that. Just let me know when you are able to see. Okay.
Yeah, yes, sir. You can continue.
All right, and I'm so sorry for the for the slight disturbance. I just changed my place now, so hopefully it should be
Yeah.
okay. Because that was coming from the roadside, so I can't help it. I can't stop that. So at least I thought I can change my position. All right. So coming back to fixed dose ratio combination preparation. So these are the few scenarios where we may have to use combination of the drug preparations. Yeah. At the same time, there are some disadvantages just for the understanding. Hopefully, because many a times there are some what do you call side effects because some hepatic or renal functions are altered, then the pharmacokinetics will definitely vary for the different components. Correct. So I think I don't want to concentrate more on this because there are more advantages than disadvantages, and that's the reason why in today's scenario, very rarely we see that single drugs are given. Always there are combinations are given. Okay, so that's why we consider that combination therapy is a better alternative. Okay, and as you all know, sometimes when you try to combine these drugs, even doctor will ask you that if you have any allergies or something, because when you try to add two components, there could be allergy or some other untoward reaction from one of the drug then also it will be detrimental. So we always prefer that we clarify that upfront before deciding the combinations. Okay, so let's move on and talk about actually in a high throughput fashion in a in vivo assay when we do how exactly they are done in drug discovery and development. So, see, in human, that is all driven by the clinical trials, which we will cover later. But when you do this same PK studies, pharmacokinetic studies in animals, the whole cycle of this PK study is as it is mentioned here. Okay, so it starts with a laboratory animal. The animal generally would be rats and mice, but sometimes based on the specific requirement, specific disease requirement, or disease target, you can choose the species. Okay, it could be rats, it could be mice, it could be rabbits, it could be hamster, or guinea pig, based on which disease target you are working on and what is going to be your efficacy model going forward for your disease target. Based on that, you can decide which species to use for PK study. So, for say for example here rats. So generally you will do it by two routes. One is intravenous. Now all of you know intravenous we always have to do because we want to identify calculate bioavailability after oral route. So one route would be intravenous, and in the second set of animals, we may give it orally. Okay. So after giving, there will be different time points which we decide ahead of time, like 0 minute before administration of the drug. We collect the blood sample, especially in rats. You can collect it from the tailwind. Okay, nowadays analysis is possible with even one drop of blood or few drops of blood. So tailwind collection can be done. Other ways, which is not preferred, but many colleges and students labs, they still do retroorbital flaxes. That is on the anterior and the posterior end of the eye. You can break those vasculatures using a glass beads, and then microcapillary tubes, and then collect the blood. So that is another way of collecting the blood. And the most accepted way of collecting the blood is by jugular vein cannulation. So there is a small surgery which you need to do, which takes around 20 to 25 minutes. And in the jugular vein, we connect the cannula, and you can then collect the blood at defined time points from the jugular vein directly after surgery. So this you have to do a day prior. So we do jugular cannulation surgery one day prior, and next day we do the actual dosing and then start our study. So 0 minute blood collection prior to administration of the drug. Then you have to administer the drug. If it is intravenous, not generally, it is like almost all the time in rats and mice, you have to give intravenous through tail vein. Okay, and oral, we give oral feeding needle using their in their mouth, and then we deliver the drug. And after say 5 minutes, 15 minutes, 30 minutes, 1 hour, 2 hour, 4 hour, 6 hour, 8 hours, and 24 hours. This is by and large more than 95% of the studies we follow these timelines. Okay, so after 5, 15, 30, 1 hour, 4 to 6, 8, 24 hours, we collect the blood. After collecting the blood, immediately we put it for centrifugation, generally 3000 rpm for 5 minutes. And after that, we collect the supernatant, which is plasma. And either you can store it in minus 80 degrees till further analysis. And if you want to analyze, then you can analyze it in a 96 well plate. So nowadays, protein precipitation ready-made plates are available with the coating. So you can put your plasma, predefined plasma into it. To that, you can add acetonitrile, okay, ice cold acetonitrile or methanol, and within that you can add your internal standard. You can vortex it, and then nowadays for a high throughput fashion, generally liquid handling units are used like Tomtec or multi-probe transfers. So that's how we take up the samples, and then we do the sample processing. Whatever process I was telling you to add acetonitrile, methanol, mixing up, shaking, all that thing can happen here. And then after that sample processing, here it is, we will cover this separately, but just for your understanding, a biological sample The processing can be done in three ways. One is protein precipitation, where we bring about protein denaturation using ice cold methanol or acetonitrile. Second way is liquid liquid extraction, where you use two immiscible solvents, organic solvents like ethyl acetate, dichloromethane, hexane, all these things we use. And third one is solid phase extraction, where SPE solid phase extraction cartridges are available, where the silica bedding is used, just like in a HPLC column. So we will discuss in detail about each of that sample processing technique later. But after that sample processing, directly the samples are loaded in an auto sampler, and from auto sampler they get into the mass spec HPLC or LC-MSMS, triple quadruple, and then you get the data which is taken in a spreadsheet. You calculate, and then you have the parameters like AUC, clearance, volume of distribution, half life, Cmax, concentration maximum, and Tmax, and your interpretation is ready. I mean, you can do interpretation based on the PK parameter. So this is the cycle of PK study in a high throughput fashion. In vivo assay we call it as, done for majority of the projects in drug discovery. It starts with dosing, blood collection, sample plasma separation or serum separation, loading them in a 96 well format, sample processing using liquid handlers, doing sample processing for protein precipitation, liquid liquid or solid phase extraction, loading the sample in an auto sampler, analysis by detection or detector as mass spec, a mass detector. You get the data, do the calculation using softwares, identify the parameters you want to concentrate on, and then compare it for different compounds, and you can decide which particular molecule is good to go further and which one you need to go back to the chemist or a formulation scientist and share your feedback with them. So this is the in general preclinical cycle of pharmacokinetic studies in a high throughput fashion. So basically, we have talked about absorption, distribution, metabolism, excretion. Toxicity is still left, which we'll cover in detail later. But in vitro assays, we have talked about in vivo experiment. So this is the cycle. You see, a new chemical entity coming out from medicinal chemist. Basically, physical properties would be first characterized. Then you need to develop the bioassay, which we are yet to cover. I'll talk about it. A cell-based or enzyme-based biological assay for biological activity determination. We have to do potency. Then we have covered this almost everything in ADME. So we do ADME characteristic calculation. We do in vitro experiments, which could be solubility, octanol solubility, simulated gastric and intestinal fluid, plasma stability, metabolic stability, or even PK study in animals. Then you analyze the samples, you get the data out of it, either in vitro or in vivo experiment. Whatever data, you interpret that, you report that, and then based on that, you go back to the chemist and they can start their effort again. So this is the general cycle of drug discovery process, where ADME fits into the play. There is a role for biological activity determination also. So from A to H, this cycle continues till you get your best lead molecule, which is optimized for good ADME properties. Okay. So this I have just given that after doing an in vitro in vivo experiment, you can always go back and if chemist has modified the structure, you can again do the bioassays for potency and selectivity. So these are all interrelated. You may have good potency selectivity, but if in vivo experiment doesn't show good PK activity or a good ADME properties, you may have to request chemist to do some structural modification. The moment he does the structural modification, again you have to do the potency assay, either in cell based or an enzyme based experiment. So this is the cycle of PKPD cycle. So here I talked about same thing in from a PK perspective, here from a PD perspective, because in vivo will give you parameters like bioavailability, Cmax, Tmax, T half. So those things are directly related to the potency of a molecule. But please remember, this potency, we are talking about in vitro potency, and here in vivo experiments, we are talking about experiments in live healthy animals. Efficacy is to the molecular target. So this could be a cell-based or an enzyme-based assay, whereas this would be in a healthy animals. Always we do our PK study. Okay. So with that, I think we come to the end of pharmacokinetics discussion. Of course, we need to still cover about the methodology. Although in brief we have talked about it, how dosing happens. But I would like to share the entire pictorial journey, like how the cannulation is done, how exactly dosing happens, and then the sample processing and all. So that we will cover in detail along with the clinical trial aspect of it. But this was in nutshell about ADME, which is PK studies in animals, when you actually do it in a pre-clinical discovery scenario. So with this, I think we have covered all the in vitro aspects, including alternatives to animal experimentation, which is using hepatocytes, which are gold standards. Then we have talked about enterocytes, which is very newer technology, which majorly focuses on metabolism in enterocytes. Then we have talked about all the in vitro assays, starting from solubility to octanol solubility, to simulated gastric fluid stability, simulated intestinal fluid stability, plasma stability, microsomal stability, and then we have talked about pharmacokinetic studies, where we have picked up most important essential pharmacokinetic parameters, and we have discussed that. And then we have also discussed about how this whole cycle works of PK in drug discovery scenario, starting from dosing all the way to analysis and then interpretation of the data. So I think I'll stop here with ADME and PK study discussion. And in the subsequent classes, we will now go to efficacy, methodology of PK, and then toxicity studies. So, as we always say that the whole aim of drug discovery and development is to have a safe and efficacious drug in market. I mean, to be able to bring a safe and efficacious drug into the market. So we will understand about the efficacy models, and we will also understand about the toxicity models. And toxicity studies, let me just tell you that they are well defined. There are regulatory guidelines. Accordingly, you have to do that, and you cannot deviate from that. So they are pretty much guideline guideline derived studies, and you cannot deviate from those guidelines. So based on where you want to submit the data, you can do the toxicity study as per the guidelines of applicable in that particular country. So that is about toxicity studies. And then we will talk about GLP, which is good laboratory practice, because exploratory environment is different in a research scenario, but when you're trying to submit the data to regulatory agency, it is expected that certain amount of data has to be generated in a GLP certified environment, in a GLP certified lab or GLP compliant environment. So we will talk in detail about GLP in the subsequent class. So with that, Maheshwari, let me stop here and would be more than happy to take any questions if you have.
Yes, sir. But time is 12 o'clock only, no? Students, anyone having any queries, please respond.
Okay, Maheshwari, no worries. If there are no further questions, then maybe we can. They can ask me questions maybe tomorrow also. That's fine. Tomorrow we have a class, right, Maheshwari?
Tomorrow, yes, sir, we have class.
Yes. And how about day after tomorrow on Friday?
On Friday, we didn't have, sir. January 1st.
Okay, we don't have. Okay,
Yeah.
all
On
right, great.
second also, we won't have.
Second is Saturday.
Saturday, Sunday.
Okay, okay. Then we'll have on Monday. Fine.
Yeah,
So,
Monday.
so then tomorrow we will in tomorrow's discussion. I'll try to cover up more content so that then there's 3 day holiday, so students can revise and refer those things, and then on Monday we can start a new topic. So tomorrow I'll try to cover more content so that for next couple of days then there are no classes. Okay. So
Yeah.
so thank you everyone for your time, and then my apologies for slight disturbance in the background, but yes, hopefully tomorrow onwards it will be again fine. So thank you everyone. Thanks a lot for your time. Thank you, Maheshwari. Thanks a lot.
Sir, but only 1 hour is completed, no?
Yes,
Yeah. Okay, sir. Thank you. Thank you so much. Today topic is completed, students. Again, we'll have section on tomorrow.
absolutely, yes.
Okay, I'll give the feedback form also to all of you. All of you submit the feedback form. Anyone having any queries, please respond. No doubts, no queries. Okay.
Okay. Thank you,
Thank you, sir.
Maheshwari. Thank you,
Thank you,
Maheshwari.
sir.
Thank you.
Students, I have given the feedback form. Please, all of you submit the feedback form. Please check it and submit the feedback form. Based on that only, we'll provide the certificates. Check it. All of you got the feedback form. It is opening. Please check and give the feedback form. I have given YouTube, Facebook, AirMeet, and yeah, ADMET, Facebook, YouTube Live also. I have given feedback form. Please check and submit the feedback form. Thank you everyone. The section is closed for today.